So, continuing chapter 5 and organic chemistry, we're going to move from carbohydrates and lipids to protein. That's not right. EI. Remember I said in the lab, I'm a visual thinker. I look at it and go, wait a minute, that doesn't check. It's bad when my brain goes, that's spelled wrong, but it won't give me the right picture to be sure. And I have to write it down to see if it spells right. So proteins are long chains of amino acids. Now, you don't need to know a lot about amino acids, but I'm going to talk about them. Amino acids, not a terribly big component. And in fact, I just realized that there's a word that goes with this that I need to deal with, that any molecule that's a long chain of the same sort of bits is a polymer. So starch is a type of polymer. It's a long chain of sugars. But it turns out proteins or polymers also literally means many bits. And nucleic acids are polymers. So you get these long chains. And we make a lot of artificial polymers too. Plastics are polymers, and a whole bunch of things that we use in everyday life are made by stringing long bits and pieces together. But you can see here that it's got a carbon in the middle with its 4 bonds, right? Carbon's got 4 bonds. On one end, it's got nitrogen. So this is one of the ones that does have nitrogen in it. That's an acid group, because under a lot of conditions, this thing can give hydrogens up to the environment. That's what makes it an amino acid. And then what makes one amino acid different from the others is what's stuck to the carbon on the side. There's a couple of different ones where sometimes you got little bitty things, and sometimes you got really long things. They have different features. As amino acids connect to one another in different ways, you can have a group that's hydrophilic and a group that's hydrophobic. Amino acids are also called peptides. That's an old word for them. And here's a few different ones. This one's pretty simple. They've got the side groups up on the top here. This one's pretty simple. This one's a little complicated. This one has sulfur in it. Some of them do. Some of them have, I think sulfur is the only other thing that you get. But there's another one with sulfur. And they're just showing them, and they connect here, right? And, dehydration synthesis. Now, there's not that many different amino acids in living things. And what makes them different is the side group. So here's one, the side group's just a hydrogen atom. Here it's just a carbon group. This one's got this weird connection. And you can see that some of them get pretty bizarre, that have a lot of things. And they all are named with INE on the end, but that's not necessarily, that's not a rule we're gonna look at. Because down here it's aspartic acid. So, and now they're calling it aspartate and glutamate. Sometimes you'll hear people talk about, there's a thing that they put in food to help preserve it that's called monosodium glutamate. This is the glutamate part. It's just an amino acid with sodium stuck on it. So for a long time, people said that they were having digestive problems with it, and it doesn't really make any sense that they would, because it's just an amino acid. Tryptophan, you hear that sometimes when people talk about being sleepy. But there's a lot of different ones. But In theory, there's an infinite number of different ones you could have. These are the only ones you really find in living things. 20 to 21 of them. If you look in some really bizarre living things, you can find a few more, but it's a limited list. Not too surprisingly, when you link them together, you link the carbon on one end to the nitrogen on the other end, an H and an OH, water comes out, and now you've got them linked together with what's called a peptide bond, because they used to be called peptides. These things have 4 levels of structure. They have primary, secondary, tertiary, and quaternary. Whoops, boy, I can't spell today. Well, they have primary, secondary, and tertiary. Some of them have quaternary, some of them don't. So the 4 levels don't apply to every single protein. Primary is simple. Amino acids are stuck in a chain. That chain has an order to it of amino acids. Okay, I'm going to throw this over the top of what I've got written, just to give you an idea. It can be represented as a long ribbon. And there's an amino acid, there's an amino acid, there's an amino acid, there's an amino acid. And so you get this order. This is how it's built, 1 amino acid at a time. And so you wind up with 1 level of structure. But they don't attach to each other randomly. When you attach one amino acid to another amino acid, here's amino acid 1, and it may attach to the next one just like this, or just like that, or just like this, or just like this. It's attaching in 3 dimensions. So it isn't just side to side, it could be side and slightly forward, or slide and slightly back, or up and slightly forward. So there's a whole bunch of different ways they connect to one another. And that produces patterns when you start to let the string exist in the real world. So much so that you'll get honest to goodness kinks. And if you get a kink and this one is going past that one, they may cross connect to each other. So you'll get patterns within the way that the string conforms. That's secondary. Since these are really, really big, they're going to be parts inside that form these patterns. Typically, there's 2 different types of patterns. You get one side going up and down and up and down and up and down and up and down because of the way that they connect to one another. And it's right next to a parallel part of the string. It went up, it curled, it came back. And it's also going up and down and up and down and up and down. And they connect to each other, and they call these pleated sheets, because that's what they look like. They look like sheets with folds in them, with pleats. Now, sometimes they connect to one another and they form a spiral. Spiral technically is called a helix. And these are called helices. That's a plural of helix. But these happen within the protein. So, sorry, this thing, that's this weird stretchy thing. So, here's a pleated sheet up on top. They have hydrogen bonds, you know, these partial charges that connect things. Hydrogen bonds holding the sheets together. Here's one of the helices. Inside a bigger protein, you've got these in some places, you've got the pleated sheets in another place. And you've got these curls and these connections, and what you wind up with on the outside is kind of a face that faces the world, a three-dimensional shape. Proteins do almost an infinite number of things because you can make them into infinite shapes that interact with the world in a bunch of different ways. Now we are to tertiary structure. I'll do it here. Overall, you get a 3D shape. Let me find what I'm looking for. Here's a bunch of different proteins. And they can be represented as bumpy blobs. This is, I've lost my pointer. This is actually an antibody that attaches to invader molecules there and there. These would be copies of each other. And when this connects, it changes the shape of the whole molecule, and this end essentially becomes an alert flag that says, hey, we're hooked onto something. Immune system cells, come do something about this. There's an activator protein that attaches to DNA. This curly Q thing is DNA. It's a lot of these things. Here's where two fit together. This is almost certainly a place where something fits in there. The shapes do things. In some cases in your muscles, you've got proteins that actually move across one another to pull your muscles shorter. You've got proteins that pull on each other at a kind of an angle to make things like a flagellum spin. So, and you've got a lot of proteins that will grab the sucrose in your Cap'n Crunch and stress it so that you can get the sugars to come apart. That's tertiary structure. Now, some of these, I'm not entirely sure which ones because you can't tell looking at the outside, some of these are more than one string. The functional three-dimensional shape actually has multiple strings in it. If that's true, you've got the fourth level of structure. And that's quaternary. So if the functional protein has multiple strings, how they fit together. So this is quaternary structure, where whatever that protein is, this is the functional protein, but it's actually several strings fit together. And they've got the secondary structure and the tertiary structure as well, but they only work when they're all locked together. Your hemoglobin is like that. Your oxygen carrying part of your blood has quaternary structure. Here we go. And hemoglobin actually has, on top of the protein structure, it actually has a little iron-containing group that has to fit into a notch in here. And the quaternary structure for hemoglobin are two, what they call alphas and two betas. So there's 4 different units that fit together, but this whole thing is hemoglobin. Here it is with oxygen attached to it, here it is without oxygen attached to it. When we talk about sickle cell, there's a problem in the primary structure. One of the amino acids is different that shouldn't be. And if this oxygen comes off of all 4 of these, the overall shape changes of the molecule. This is typically a pretty roundish molecule. Sickle cell with the oxygen removed becomes long and pointy. The whole outside of it changes. If you have a red blood cell full of that stuff, the cells become long and pointy, sickle cells, and that's a problem. We'll talk about that later. So you're trying to show people how these things are put together. This is actually showing the bonds and stuff. And a graphic like this is not really useful except on a computer where you could tell it to turn. And then you can actually see what's going on. If you took this, this is just a whole mess. You're looking through the molecule and all the different bonds. But if you could turn it, you could kind of see how it all works. This is simplifying the structure to show the helix formations, the secondary structure, and in some places there'll be a flat arrow, and a flat arrow right there too is supposed to be one of those pleated sheets. That's just the way they represent them. And then you just show the outside of it. You show the outside of it, and it's easier to see something like this is probably the active site for an enzyme, where the enzyme fits onto some other molecule to make it do something. And so you've got this outside face that it shows to the world. Now, what we're dealing with proteins is fairly complicated. So I'm going to do the proteins in two separate videos. So now we've gone through the structure. Next, we'll deal with the function.